that may need energy. One of those energy requiring portions is the active transporters within the cell membrane that we learned, which need to work against the concentration gradient in order to move things across the cell membrane. And again, like I said, which type of molecules are broken down? All food molecules, which can include stuff like carbohydrates, lipids, and proteins, they can be broken down, processed to make ATP. And every single living organism, plants, fungus, algae, protists, prokaryotes, they all carry out cellular respiration. Viruses do not carry out cellular respiration, which is why, again, one of the reasons we do not consider them to be living organisms. And now, when we get to cellular respiration, we see that there are, well, if you look at the reaction at the bottom, this is the basic reaction. The reactants that we're using is glucose and oxygen, and the end products that we get are carbon dioxide, water, and ATP. However, in the middle of this reaction, there are also intermediate products. The glucose, the C6H12O6, is not directly broken down to form ATP for the most part. There are also other products that are formed, pyruvate, pyruvic acid, acetyl coenzyme, etc., etc. Stuff that is formed in the middle, which are then further broken down as necessary. Those parts are known as the intermediates of this metabolic reaction, as we learned about what a metabolic reaction is. So, when we look at the stages of respiration, you can see here there's four parts shown: glycolysis, the pre-Krebs cycle, the Krebs cycle itself, or citric acid cycle, and the electron transport chain, or ETC. Of these parts, you can consider there to be three major portions: the glycolysis, point 1, Krebs cycle, point 3, and electron transport chain, point 4. Point 2, which is the pre-Krebs cycle portion, is just one specific reaction that happens in between step 1 and step 3. So before I look at each of these steps in detail, which is the main goal of today's lecture, do you have any questions so far about what we learned in regards to just the initial part of respiration?
Not sir.
আচ্ছা,
No, sir.
ঠিক আছে, তাহলে let's carry on. যদি এখনো question না থাকে কারো, before we break down each of those steps, we need to understand যে respiration itself কিন্তু দুইটা ভাবে আগাতে পারে। একটা হলো aerobic, একটা হলো anaerobic. So, আমরা যখন রেসপিরেশনের কথা বলি সেলুলার, তখন আমরা ইউজুয়ালি মিন করছি কিন্তু aerobic respiration। মানে আমরা যে রেসপিরেশন রিঅ্যাকশনটা দেখছি, we see that oxygen is being used in this portion. তো অক্সিজেন না ইউজ করলেও কিন্তু আমরা respiration carry out করতে পারি। It's possible, even though it isn't usually done. আমাদের শরীরের মধ্যে যে চারটা স্টেপের কথা বললাম আগে, the first step mentioned here, glycolysis, is the only step যেটা anaerobic ভাবে হতে পারে। Glycolysis can occur both aerobically and anaerobically, এবং depending on এটা aerobically না অথবা anaerobically হচ্ছে, মানে depending on whether glycolysis is using oxygen or not using oxygen, there are different products and different ways in which the reaction progresses. Another point to note is that the first step, again, glycolysis, is the only step that is happening outside of the mitochondria. This part of respiration takes place or begins in the cytoplasm of a cell. After this step is concluded, the remainder of the steps then happen within the mitochondria. As we know, mitochondria is where respiration usually happens, but not all of respiration happens within the mitochondria. It's only the first step that happens outside of the mitochondria. Usually from each of these steps, be it glycolysis or Krebs cycle or electron transport chain, there are two important types of products formed: an electron carrier or an energy carrier. Again, as we already learned, energy carriers are a molecule that is made which can carry energy that is made whenever a reaction of respiration happens. However, another type of molecule that's important to note are electron carriers. What are electron carriers? Electron carriers are simply a type of molecule which can help to transport the loose or the spare electrons that are released whenever a reaction happens. Because keep note, whenever you break down a molecule, it's not just the chemical bond energy that you're releasing. Oftentimes, you're breaking down a big molecule into a smaller molecule, and this can mean that there's some electrons lost. For example, if you have a molecule with 25 electrons, and then you're breaking it down to make two molecules with 10 electrons each, there are 5 electrons that are released from the process that have nowhere to go. These electrons are then carried using the electron carriers such as NADH and FADH2 to other parts of the reactions, which may need spare electrons. So remember that usually there's two types of things released: energy, which is carried by ATP, and electrons, which are carried by NADH and FADH. This diagram on slide 21 is just an overview or overall image of all the different steps that happen. We can come back to this, or you can look at this after you've understood each of the individual steps. So let's go to glycolysis, or the first step, as we talked about. Yes. Oh, sorry, Josh. Okay. Now, let's start with step one of respiration, or the first step. The first step of respiration, as we know, is glycolysis. And when we consider this respiration to happen aerobically or with the help of oxygen, what happens is the main reaction is, we look at the bottom right section, the breakdown of one molecule of glucose into two molecules of pyruvate. What is happening is a 6 carbon molecule, which is the glucose, is being broken down into two 3 carbon molecules. And if we look at this, it is the breakdown of a larger molecule into two smaller molecules. This is a catabolic pathway or a catabolic reaction that is happening here. The larger molecule is being broken down into two smaller molecules. So the reactant is glucose, and the product for this particular step is pyruvate. But for all of respiration, pyruvate is an intermediate product because we're going to break down pyruvate further in the next step. Also note here that if you look at the image above or look at the product, it's not just two pyruvates that are released from glycolysis. As a product, you're also getting two molecules of ATP and two molecules of NADH. There is some energy released from the process because, again, we know that a catabolic reaction releases energy when you break down a big molecule into a smaller molecule. You're getting two molecules of ATP because the ATP is needed to carry the energy that is released, and you're getting two molecules of NADH because you need the NADH to carry the electrons that are released. One important point to remember, or at least something you should remember that's not written here, is that even though it's written that the net yield is 2 ATP, what does the word net yield mean? Net yield কথাটার মানে হলো তোমার প্রফিট কত লাস্টে, যে তুমি কয় মলিকিউল লাভ হিসেবে পাচ্ছ। এখানে লেখা আছে যে you're getting two molecules of ATP as the net yield. কিন্তু ultimately actually কিন্তু glycolysis থেকে আমরা 4টা molecule of ATP পাই. So why is it not written like that? It's not written like that because to initiate ATP, sorry, to initiate glycolysis or to start the process of glycolysis, আমাদের two molecules of ATP দরকার হয়. So if you need two molecules to start and you're getting four molecules back from the reaction, how much of a profit are you having? You're having a yield or a profit of 2 ATP. That's why it's written here in a simplified form that ultimately from the process of glycolysis, you're getting a product of 2 ATP. As long as you remember that, you're good enough. So you're getting two molecules of ATP from the process, along with two molecules of NADH and two molecules of pyruvate by breaking down one molecule of glucose. কারো কোনো কোয়েশ্চেন থাকলে হাত তুলতে পারো, আর যদি কোয়েশ্চেন না থাকে, in a few seconds I'm going to move on to the next step. Arup Das.
Sir, what was the primary product of glycolysis?
The primary product or the product of glycolysis is pyruvate. এই যে এখানে দেখছো একটা মলিকুল, one molecule of glucose 6 carbon molecule কে তোমরা দুইটা 3 carbon molecule ব্রেকডাউন করছো। each of these three carbon molecules is known as pyruvate.
Oh, okay.
এই যে ডান দিকে দেখছি pyruvate, Pyruvate. The pyruvate is a simpler form of the glucose molecule.
Yes, sir, understood.
Okay, good. আর কোনো question? Okay. Now থাকলে let's move on to the next step. The pre-Krebs cycle. So আমি যা বলেছিলাম যে all that's happening in the pre-Krebs cycle is one particular reaction. Excuse me. Sorry. So what is happening in this one particular reaction? এই রিঅ্যাকশনটা হলো আমাদের যে পাইরুভেটটা পেলাম আমরা, the pyruvate that we just got from glycolysis, this cannot be used in the Krebs cycle. এটাকে ইউজ করার আগে this needs to be processed and turned into something else. So what's happening here is that this pyruvate is reacting with an enzyme or combining with an enzyme called coenzyme A or CoA for short. This CoA enzyme combining with pyruvate produces a molecule of acetyl coenzyme, acetyl CoA. So the reaction is pyruvate plus CoA forming acetyl CoA. And as you can see here, acetyl coenzyme A is a molecule produced by almost all nutrients. When all types of organic molecules, gyral process করে না কেন glycolysis থেকে Krebs cycle ঢোকার আগে সেটাকে coenzyme A এর সাথে combine করে acetyl coenzyme A বানাতে হবে. And then once that happens, that acetyl coenzyme A can enter the Krebs cycle. So this is the intermediate reaction in between glycolysis and Krebs cycle. Now let's look at Krebs cycle, which is probably the most complicated part of respiration. Again, as we know, Krebs cycle is a fully aerobic reaction or a reaction that needs oxygen. And the important thing to remember here in the Krebs cycle is that there are two things happening. A molecule is being formed and a molecule is being broken down. So what's being formed? So the acetyl coenzyme A that we just talked about is combining with a 4-carbon molecule to make a molecule of citric acid. If you look at this Krebs cycle diagram, again, you don't have to remember diagram, but it's good to help in order to help you understand. So you see here at the top that the acetyl coenzyme A is entering the Krebs cycle. And then as it enters the Krebs cycle, to the left, you see a 4-carbon molecule. This 4-carbon molecule combines with the acetyl coenzyme A to form a 6-carbon molecule. This 6-carbon molecule is known as citric acid. That's the formation part of the Krebs cycle. And then the breakdown part of the Krebs cycle is this citric acid that just formed is then broken down into carbon dioxide. See here, this citric acid 6-carbon is broken down into ultimately carbon dioxide and other carbon chain products. Those carbon chain products then reform with the acetyl coenzyme A to form more citric acid. See how it's cycling. This is broken down, this is broken down, this is broken down, and then it goes back, reforms the acetyl coenzyme A, on and on and on. That's why we call it a cyclic process. Because whatever product is formed from the breakdown of citric acid, other than carbon dioxide, which could be the 4-carbon molecule, goes back and reacts with acetyl coenzyme A to again form citric acid. So this carbon dioxide ultimately is one of the big products of Krebs cycle. Other than carbon dioxide, we're also getting other parts like ATP, NADH, FADH2, energy carrying molecules, and electron carrying molecules. So as you can see here, again, according to the diagram, the products that we're getting are ultimately carbon dioxide, ATP, NADH, and FADH2. And how much ATP are we getting from this reaction? You need to remember that here it's written that it produces, you can see here that 2 ATP molecules from 1 glucose molecule. How does that happen? Because each cycle produces 1 ATP. And what does each cycle process? Each cycle processes one molecule of acetyl coenzyme A. What formed from one molecule of acetyl coenzyme A? One pyruvate. And we know that we get two pyruvate from each glucose in glycolysis. Thus, one molecule of glucose will have two molecules of acetyl coenzyme A, and each of them produces one ATP. So together, one molecule of glucose, which is the reactant all the way at the beginning of the process, will lead to you getting two molecules of ATP from the process. However, if I were to ask you how much ATP is produced if I were to react one molecule of acetyl coenzyme A in the process, then you would say that you would get only one molecule of ATP. Do you understand this difference?
Yes, sir.
সবাইকে হ্যাঁ এটা একটু নিয়ে একটু বুঝে নেও যে এটা নিয়ে কোশ্চেন থাকলে আবার জিজ্ঞেস করতে পারো আমাকে যে কেন একটা এটিপি বলি আমরা যখন একটা অ্যাসিটাইল কোএনজাইমে কিন্তু পুরো প্রসেস থেকে কেন আমরা বলি যে দুইটা এটিপি পাচ্ছি কারণ পুরো প্রসেসে আমরা আলটিমেটলি দুইবার করে এই অ্যাসিটাইল কোএনজাইম এটাকে প্রসেস করছি কারণ একটা গ্লুকোজ থেকে আমরা দুইটা পাইরুভেট পাচ্ছি যার থেকে আমরা দুইটা অ্যাসিটাইল কোএনজাইমে পাচ্ছি যার জন্য আমরা ক্রেবস সাইকেলটাতে দুইবার ঘুরছি যে টার্নস টুয়াইস per glucose molecule. So ultimately আমরা এখানে প্রোডাক্ট গুলো পাচ্ছি from Krebs cycle is NADH, FADH2, ATP and carbon dioxide. Then let us look at the last step of this process, electron transport chain. So this electron transport chain is a series of proteins in the inner mitochondrial membrane. And what does this do? This is where most of the ATP that we need from the reaction is produced. আমাদের যত এটিপি দরকার হয় বেশিরভাগই কিন্তু এই ইলেকট্রন ট্রান্সপোর্ট চেইনে বানানো হয়। এই ইলেকট্রন ট্রান্সপোর্ট চেইন যা করছে সেটা হলো এই যে এতক্ষণ আমরা বললাম না যে NADH বানানো হচ্ছে, NADH, FADH2 বানানো হচ্ছে from glycolysis, from Krebs cycle. All of the electron carrying molecules that we got, those are then carried forward and moved here to this part of the process, to the electron transport chain. এই যে লেখা আছে, the third point. As the electrons move down the chain, energy is released, and this energy is used to move hydrogen protons out of the matrix. And these hydrogen protons are obtained from water. এখানেই হলো আমাদের এই যে বলি না আমরা রিঅ্যাকশনে ওয়াটার কেন দরকার? This electron transport chain is where the water is needed. So the water that we have within our bodies, that water contains hydrogen ions, H plus, আমরা জানি. So that hydrogen ion reacts with all the electrons that are carried forward from the previous steps. যতগুলো NADH, যতগুলো FADH2 আছে. All of that reacts with this hydrogen ions to produce energy and then have oxygen as working as a final electron acceptor at the end of electron transport chain. But ultimately, এই রিঅ্যাকশনটা হওয়ার পরে যা ইলেকট্রন আমাদের অ্যাকসেপ্ট করা লাগে or the electrons, the free electrons এতক্ষণ যে ঘুরছিল, all the electrons that are being carried by NADH and FADH2, those are then reacting with these hydrogen ions and ultimately being accepted by oxygen. অক্সিজেনই আমরা ফাইনাল ইলেকট্রন অ্যাকসেপ্টর হিসেবে কাজ করতে পারি। সো এখানে এই যে আমরা বললাম ইলেকট্রন গুলো রিঅ্যাক্ট করে ওয়াটারের সাথে এনার্জি গুলাকে রিলিজ করছে, এই এনার্জি দিয়ে আলটিমেটলি আমরা তাহলে কতগুলো এটিপি বানাতে পারি? We can make 32 to 34 usable energy, usable molecules of ATP from the electron transport chain. And we're also getting water as a byproduct in this part. So, from cellular respiration, আমরা টোটাল প্রসেস থেকে কতগুলো এটিপি পাচ্ছি? গ্লাইকোলাইসিস থেকে আমরা দুটো এটিপি পাচ্ছি, ক্রেবস সাইকেল থেকে আমরা দুটো এটিপি পাচ্ছি, and electron transport chain থেকে আমরা 32 to 34 ATP পাচ্ছি. So you can see where the majority of the energy is made in respiration. Most of it is made during the electron transport chain. So you can see here that ultimately the 36 or 32 ATP পাচ্ছি, most of that is being made during the electron transport chain. Any questions, anyone? আচ্ছা, কোনো question নেই? ঠিক আছে, the last thing that we're going to talk about today is going back to the aerobic and anaerobic respiration point. আমরা যে বলেছিলাম যে glycolysis is the only part of the reaction where reaction can happen aerobically or anaerobically. Aerobically হলে তো we process normally, we progress to the Krebs cycle, to the electron transport chain, so on and so forth. কিন্তু যখন anaerobically হয় তখন এই রিঅ্যাকশনটাকে আমরা বলি fermentation. What is fermentation? Fermentation is a reaction of breaking down glucose which is making ATP without using oxygen. যেহেতু আমরা অক্সিজেন ব্যবহার করছি না এই পার্টে, আমরা অন্যান্য স্টেপ গুলোতে আগাচ্ছি না। We're not going the Krebs cycle, we're not going to the electron transport chain. Whatever energy we're getting, we're only getting from glycolysis. শুরু হয় গ্লাইকোলাইসিসে, শেষও হয় গ্লাইকোলাইসিসে। তো একটা এক্সাম্পল আমরা দেখতে পারি এখানে যে দেওয়া আছে, ল্যাকটিক অ্যাসিড ফার্মেন্টেশনও একটা এক্সাম্পল, অ্যালকোহল ফার্মেন্টেশন একটা এক্সাম্পল। এই ল্যাকটিক অ্যাসিড ফার্মেন্টেশনটা কিন্তু আমাদের শরীরের মধ্যে হয়। তোমরা যখন অনেক এক্সারসাইজ করো, তোমরা যখন দৌড়াতে থাকো, তোমার টায়ার্ড লাগে কেন? তখন টায়ার্ড লাগে কারণ আমাদের বডি যখন অনেক এক্সারসাইজ করছে তখন তারা এরোবিক রেস্পিরেশন করতে পারে না, তখন তাদের অ্যানারোবিক রেস্পিরেশন করতে হয়। আর যেহেতু তারা অ্যানারোবিক রেস্পিরেশন করছে, আমাদের মাসলের মধ্যে ল্যাকটিক অ্যাসিড বিল্ড আপ হচ্ছে। এই ল্যাকটিক অ্যাসিডটা হলো স্লাইটলি টক্সিক, এটা হলো আমাদের শরীরের এই ব্যথা বেদনা বাড়ায় দেয়। দ্যাটস ওয়াই আওয়ার মাসলস হার্ট, দ্যাটস ওয়াই উই ফিল টায়ার্ড, because this anaerobic respiration is happening within the body. And ultimately, এই যে দেখছি আমরা ল্যাকটিক অ্যাসিড ফার্মেন্টেশনে কি হচ্ছে, what's happening here, দুটো একই ডায়াগ্রাম, বাট জাস্ট ডায়াগ্রামটা হচ্ছে ইম্পর্টেন্ট মনে রাখবো তোমরা, যে শুধু গ্লাইকোলাইসিসের মধ্যে আমরা, it's just the 2 ATP. Remember that we got 2 ATP at the start of glycolysis. That 2 ATP is all of the energy that you're getting as a product. So CoA 2 ATP or CoA 36 to 38 ATP from the whole aerobic process. Thus, anaerobic process is not only less efficient, it also produces a lot less energy ultimately. This slide, number 33, shows you the differences between aerobic respiration and fermentation. তোমরা দেখতে পাচ্ছ যে এখানে ডিফারেন্স কি, এটা তোমরা পড়ে রাখো যে how is aerobic respiration different from fermentation. And lastly, fermentation আমরা inefficient বললেও it has a couple of advantages. First advantage হলো যেহেতু এটার অক্সিজেন দরকার নেই, it can allow organisms to survive in places where there is very little to no oxygen. It increases habitat. It lets organisms live in places where there is little to no oxygen, like deep water, soil মধ্যে, digestive tract মধ্যে। যেসব জায়গায় অক্সিজেন সহজে না পেতে পারে সেখানেও life can survive. And the second advantage is the speed. কারণ শুধুমাত্র গ্লাইকোলাইসিস হতে যত টাইম লাগে, that is a lot less than glycolysis, the Krebs cycle, the electric transport chain. So fermentation can happen a lot quicker than other processes of respiration. However, ultimately aerobic respiration কিন্তু টোটাল এনার্জি অনেক বেশি দিবে তোমার ফার্মেন্টেশনের চেয়ে, much more ATP than fermentation. ঠিক আছে, any final questions? That was it for lecture number 7 in general. Last যে দুটো diagram আছে, those are just giving you this is just giving you an example of the both processes. So if you have any questions, please ask me now. If not, we can begin the quiz number 2. Okay, question থাকলে জিজ্ঞেস করতে পারো। In the meantime, let me pull up the Google form for you to answer, please.
স্যার ফর্মে দেই।
আমি তোমাকে ফর্মের লিংক দিচ্ছি।